おはようございます。では始めさせていただきます。Okay, let's get started. This is Yuri from湘南アイパーク. Thank you for joining us today from Toronto and湘南アイパーク and around the world. And this virtual pitch event is a joint event of JETRO Toronto and湘南アイパーク. And this is a monthly event we are trying to that discusses a different Canadian region each time and invites local startups who are interested in working with Japanese pharma, life science companies. And today, this is our first event focusing on the Toronto ecosystem. So to give the opening remarks, I'd like to introduce Toshio Fujimoto, General Manager of湘南アイパーク. Fujimoto-san, can you start your presentation?
Sure. So good morning, everybody, or good evening, everybody, and welcome to the first joint program with JETRO and湘南アイパーク. So could you go to my slide? Yes. Let's see the beautiful facade of our facility,湘南アイパーク. And please feel as if you were here at iPark today in this event. So iPark started 2.5 years ago as the first a science ecosystem in Japan in the field of life science and biotechs. It is a huge research center, with 2 million square feet and with all the facilities for drug discovery, vivarium, bioexperiments, and so on. So please come once the coronavirus, this pandemic will be resolved. So please to go to the next slide. Yes. So we have currently, since our launch in 2018, 2 years ago, we have evolved into a dynamic community of nearly 100 global companies and 2,000 innovators, researchers, like Takeda with 700 researchers, and Tanabe Mitsubishi with 250 researchers, and so on. So that please see the variety of our tenant companies, resident companies here. So we are taking a so-called mixed model approach. Mixed model. We mean that diverse life science players are gathering from small startups to large pharma and academia and governments. And they are gathering to collaborate, innovate, and accelerate. And today, some of these companies here at iPark joining to look for potential partners, partnering with you. And next slide, please. Yes. So iPark will become a gateway for global companies from and to Japan. I mean, for companies entering Japan and Japanese company to expand globally. We are partnering with global incubators like here, like Central, TIM and Taiwanese Research Park to make connections between global startups and Japanese companies. So in this context, we are today collaborating with JETRO to facilitate new connections. I hope you will find new business opportunities today. Thank you very much.
Thank you, Fujimoto-san. And then I would also like to introduce Sakai Takuji-san, Executive Director of JETRO Toronto. Sakai-san, can you start your presentation?
Thank you very much for kind introduction, Ms. Ogiso. Good morning in Japan and good evening in Canada. Thank you very much for attending Discover Life Science Canada. The Japan External Trade Organization, JETRO, is a government-related organization promoting mutual trade and investment between Japan and the rest of the world. Recently, we focus on promoting investment into Japan, expanding agri-food exports, and supporting overseas market access for medium and small-sized Japanese enterprises under the growth strategy of Japanese government. JETRO Toronto is keen to assist innovative Canadian companies interested in investing and bringing their unique technologies and business models into Japan. So it is important to provide information to all Canadian companies regarding the Japanese market as an investment destination. Since October 2018, we have held a series of reverse pitch events in key innovation ecosystems across Canada called Generating Innovation with Japan. we call GITCH, providing business opportunities between Japanese companies that are conducting open innovation and Canadian startups that have advanced technologies and solutions. As a result, more Canadian startups are considering entering Japan. Last February, we had GITCH specified for health tech in Toronto. Japanese pharmaceutical companies made presentation for Canadian startups, and after that, they had one-on-one meeting. Because there are many healthcare and pharmaceutical startups which have innovative technologies in Canada. After the pandemic of COVID-19, we could not have those types of events. But fortunately, Mr. Tyson Gaby, Associate Director at JETRO Toronto, who is MC today, got to know with some of the staff of湘南アイパークat Bio Online Trade Show. Then we get the opportunity to introduce some of the Canadian innovative startups to Japanese companies. I hope you can find the interesting startups and start the business with them. Thank you very much.
Thank you very much, Sakai-san. Okay, so let's move on to discuss the Ontario life science ecosystem. And before that, I want to introduce Tyson Garb. He is Associate Director of JETRO Toronto, who organized this event together. Tyson?
Hi, welcome to the webinar. So my name is Tyson Garby. I work in the Toronto office of JETRO. I've been there for over 20 years and been looking after life sciences the whole time I was there. So I'm really pleased to be able to put this event on and showcase some very interesting and innovative life sciences companies. Yuri, thank you very much for all your cooperation, and we look forward to this cooperation to put this event on and then also future events. In terms of what we have today, actually all three companies participated in the event that Mr. Sakai mentioned, this Generating Innovation with Japan event in Toronto that we did in February. We had 9 Japanese pharmaceutical companies, the digital tech companies, reverse pitching to an audience of about 70 startups in University of Toronto's incubator center. It was a really successful event. We did lots of meetings, and we also brought the Japanese companies to meet many of the ecosystem players. So the purpose of the event is really to showcase the 3 companies that we have on today, and also to inform about the ecosystems as well across Canada, the life science ecosystems. There's a lot of innovation coming out of here, and I think you may find potential partners to work with. So to start things off, we're going to have a presentation from Jenny G. She is a Senior Sector Advisor for the Ontario government in the Life Sciences team. And she's going to give an overview of Ontario, the province of Ontario, the life sciences capabilities, and also some of the specific strengths of the Toronto region. So Jenny, I'll turn it over to you.
Thank you, Tyson. So hi everyone, thank you for joining us. My name is Jenny. I've worked in the Ontario government since 2016 in various ministries, and today I'll be presenting a Life Sciences Ontario investment landscape deck. So next slide, or rather I guess first slide, next slide. Thank you. So who are we? We are one of the 24 ministries of the Ontario government. So essentially we are a one-stop shop for international investors who want to invest in Ontario. Next slide, please. What services do we provide? We provide services such as business intelligence, immigration advice, site selection, customized tours, government incentive programs, and referrals to professional services. So feel free to reach out to our office. Next slide, please. What is Ontario? We are Canada's most populous province with 14 million people who speak over 200 languages and who come from countries all over the world. We're also Canada's economic engine. We make up 39% of Canada's GDP, 46% of goods produced in Canada, and 42% of total exports. We have Canada's largest international airport, which services 175 different international destinations. And if you decide to invest in Ontario, you'd be in good company because over 63% of foreign companies in Canada have a presence in Ontario. Next slide, please. As a trading nation, Canada is the only G7 country in the world with free trade agreements with all other G7 countries. We have 51 FTA signed with countries across the world, comprising of 1.5 billion consumers. And Canada and Japan are also partners in the CPTPP since 2018. We have free trade agreements signed with the European Union, South Korea, and Latin American countries such as Peru and Chile. We also have more FTAs under negotiation with countries such as India and the Caribbean. Next slide, please. And a large advantage of being in Ontario is our proximity to the USA. We have access to more than 143 million customers within a day's drive. This makes Ontario a strategically placed location for businesses that want to access the world's largest pharmaceutical market. So Ontario can be your gateway to the world's markets and customers. Next slide. So on this slide is an illustrative example of the after-tax cost of R&D investments and expenses for small, medium-sized manufacturers in Ontario. So if you conduct in-house R&D and expenses in Ontario, you can have your expenses reduced by up to 50%. So I've included a longer list of government incentive programs in the appendix for this presentation. So for your reference, but I won't go into all the details here. So next slide. So Ontario not only has advantages to international trade and government incentive programs, we also produce a steady supply of science talent. We graduate over 49,000 STEM graduates each year from 44 universities, and 40% of Canada's top 10 universities are located in Ontario, which includes the University of Toronto and McMaster University, which is well renowned for its health sciences program. So next slide. So I'll talk more about the life sciences sector, which I think you'd be most interested in. So Ontario has Canada's largest life science cluster with over 50% of total life science activity. And our sector has over 1,900 companies employing over 66,000 people. We generate $53 billion in revenue each year and export $9 billion annually. And our research is world-class. We have 23 research hospitals and institutions doing $1.6 billion in academic research each year. And every top 10 pharma company in the world has done clinical trials in Ontario. So not only is Ontario Canada's largest life sciences ecosystem, we're also attracting the largest number of VC funding, so venture capital funding in Canada. Toronto, Ottawa, and the Waterloo-Kitchener area are in the top 10 locations for VC funding in 2019, with $1.1 billion invested in the life science sector alone. Next slide. So we have a comprehensive and vibrant life sciences ecosystem. Our subsectors include the pharmaceutical sector, medical technology, and emerging fields such as biotechnology in regen med, so regenerative medicine, and the convergence of tech and science. Top companies like Sanofi, GSK, Pfizer, Amgen, and Bayer all conduct research here. Ontario is also the Canadian HQ for companies like J&J, 3M, and GE Healthcare. Our world-leading research hospitals include Toronto General Hospital, which is recognized as among the world's best 10 hospitals, in the same group as the Mayo Clinic and the Cleveland Clinic. And Ontario has always been a big supporter of innovation. We have 49 incubators and accelerators, more than New York City, London, Berlin, and Tel Aviv. And J&J opened its first J-Labs outside of the USA in Toronto in 2016. Next slide. So Ontario is attracting large-scale foreign investment as well in our pharmaceutical sector. One of our most recent investments is by Bora Pharmaceuticals, which is the world's third largest CDMO manufacturers with operations in 70 countries, and it's headquartered in Taiwan. And in March, Bora chose Ontario as the site of their North American headquarters. So this state-of-the-art manufacturing facility will employ over 400 people and will manufacture products for the entire North American market. Next slide. Ontario is also one of the world's leading locations for regen med and personalized medicine. We have over 300 regen med researchers with networks across the globe. And since 2007, the McEwen Stem Cell Research Institute has been a world leader in stem cell research. And its director, which is Dr. Gordon Keller, is renowned for his research in pluripotent stem cells. We also have the Center for Commercialization of Regenerative Medicine, CCRM, which is a public-private research consortium whose mission is to commercialize cell and gene therapies across the world. They also operate several cell manufacturing facilities in downtown Toronto. Next slide. If we talk about technology, Ontario is at the forefront of innovation. We have 300 AI firms, and we are North America's second largest IT cluster. So the Vector Institute opened in 2017 and is training the world's best AI talent. Dr. Jeffrey Hinton, who is considered the godfather of deep learning, is based in Toronto at the Vector Institute and the University of Toronto. We have always and are continuing to invest in AI. In March 2019, the University of Toronto announced a $100 million project for a newly constructed research center with 750,000 square feet of space right in the heart of downtown Toronto. And this center will anchor AI and biomedical scientists to advance biomedicine. And global companies have taken notice of our AI talent as well. So Fujitsu opened its first Canadian research center in Toronto, and LG Electronics signed a research partnership with the University of Toronto in 2018. Next slide, please. So this slide just gives you a very small snapshot of all the action that's been happening with Ontario's homegrown companies who are at the forefront of converging AI and life sciences. So Cyclica will be speaking to you today, and it's great that they're here. We also have companies such as Deep Genomics, which announced a drug candidate for Wilson disease and after using an AI platform to find it in just 18 months. And BenchSci is another Ontario company that was founded in 2017, and they're already backed by Google's Gradient Ventures, which is its first investment outside the USA. So next slide, please. So I just wanted to give this slide to show that we also have a heavy digital health presence. So we have over 300 digital health companies. Blue Dot is one notable company because they actually use AI to predict the COVID-19 outbreak before the WHO. Next slide. And we also are going through healthcare reforms, which present an opportunity for international companies who want to invest in Ontario. So we are moving away from a siloed healthcare system to a healthcare system which places the patient at the center. We are also modernizing the Personal Health Information Protection Act, which regulates digital health information to promote the use of digital health. And we have recently launched in July 2020, the Ontario Health Data Platform, which will allow researchers to better access healthcare data held by the Ministry of Health to help with our COVID-19 response. Next slide, please. So if you have any questions about Ontario, please feel free to reach out to our team. And our direct contact information is listed on this slide. We're always happy to have a meeting to chat. You can also follow us on Twitter, LinkedIn, or find us on our website. Thank you so much.
Great, thank you, Jenny. It was a great overview. Not a lot of time to explain a lot of different points. And I know that in the future, some of the webinars that we're planning to do will delve deeper into some of the other cities in Ontario and other research groups in Toronto as well. So I'm sure if you join the series of webinars, you'll learn more and more about the life sciences ecosystems in Ontario. Jenny, I believe you are able to share the PowerPoint slides afterwards.
Yes, for sure. So you can share my slides. I've given it to you in PDF, and the appendix has a lot of good resources as well. Okay, great. Yeah, and I just want to mention, it was mentioned in the chat, just reemphasize that if you want to ask questions, please write it in the chat box. We appreciate it if you could write the questions in English. But if you want to do in Japanese, we can maybe translate them later and pass them on to the companies. But we may not be able to do translation during the webinar itself. Okay, so now I'd like to move on to the first company speaker. So Cyclica, and Nahid Kurji is the co-founder, president, and CEO of Cyclica. And he has created a company that's on the leading edge of AI drug discovery, as you saw, as one of the highlights in one of the slides that Jenny had posted. And he has created, he's created partnerships with both pharmaceutical, but I think it's quite a unique business model. He's also looking to create new companies in cooperation with startups and research institutes in Japan. So I really look forward to hearing his presentation. Nahid?
Yes, thank you so much for the introduction. Can we go to my slides, please? Wonderful. So first of all, thank you very much to the organizing team at Jetro for the invitation, for putting this together, and for everybody for joining this morning or this evening, wherever you are in the world. As Toshio-san mentioned in his opening remarks, to imagine yourself at the Shonan High Park. Well, I've had the privilege over the past 2.5 years of coming to Japan about a dozen times and visiting the湘南Park close to 6 times over the past 2 years. It's a beautiful facility. And so I am quite literally imagining myself there today. And I look forward to hopefully meeting many of you in the near term. So my name is Nahid Kurji. I'm a co-founder, president, and CEO of Cyclica, a Toronto-based biotech company that is using AI and computational biophysics to transform the way in which medicines are discovered for all diseases. Next slide, please. We have built a drug discovery platform that is combining computational biophysics, structure, and AI, machine learning, and deep learning together in an integrative way. Next slide, please. As I mentioned, Cyclica is a Toronto-based company. We're close to 40 employees today. We've raised now close to $40 million in venture financing over the past 6 years, including closing our Series V round of financing in June 2020, led by Drive Capital, one of the largest venture capital funds in the US that is investing heavily in Canada, as well as Chiesi Pharmaceutical, one of our R&D partners based out of Italy. Cyclica has been recognized as one of the top 100 AI companies by Analytics Insight, as well as by CB Insights. We've been recognized as one of the top 20 AI-based drug discovery companies, and we were recognized as the most innovative AI-based drug discovery company in Canada in 2020. We have a team of computational biophysicists, computational chemists, a group of medicinal chemists, biochemists, experimentalists, and a phenomenal team of data scientists and software developers. And if you're ever in Toronto, please, make sure you get my contact information. We'd love to visit with you and spend some time. And if you're interested, next time we're in Japan, which hopefully is not too far from now, we'd like to meet you as well. Next slide, please. Now, computational chemistry has been around in the pharma industry for 4 or 5 decades. And when you think about the application of computer-aided drug design, whether it be the structure-based approach or the AI-based approach, traditionally it's focused on screening or designing small molecule ligands for one protein target. It's very much a target-centric virtual screening approach that takes a target in mind to find the best molecule that is predicted to bind or interact with that one given target. Next slide, please. Now, the challenge with the classical way of computational chemistry is that when a molecule, a ligand, is placed into a more complex biological system, a cell, animals, or humans, a given molecule is interacting with upwards of hundreds of different protein targets. And so we thought about a polypharmacology approach to drug discovery. Instead of finding a molecule to interact with one protein, the question that we asked ourselves was, is it possible to design molecules to interact with the right protein targets and avoid the wrong protein targets? Is it possible to uncover the polypharmacology of a small molecule and to leverage that to design better medicines? And that question has been the basis of Cyclica's existence for the past 7 years. Next slide, please. And on the back of that philosophy, we have built 2 first-in-class drug discovery technologies that combined are our drug discovery platform. First is ligand design, a drug design platform. Next slide, please. With ligand design, we are able to design new molecules to interact with one or multiple preferred protein targets while avoiding one or multiple undesirable anti-targets. We do this by exploring all of chemical space through a fully generative approach, through a semi-generative approach, exploring the enamine real space to get molecules that are more synthetically tractable, and through library screening to screen a set of preferred catalog of molecules that may exist internally at a pharma company. And so we're able to go all the way from a fairly closed catalog of molecules all the way up to fully generative within ligand design. In addition, we're able to design molecules so that they have the right physical, chemical, and pharmacokinetic properties. Next slide, please. And the way in which ligand design works is that it takes a seed molecule as an input, as well as a panel of protein targets, and it takes a more evolutionary, genetically inspired approach to fragment molecules and to recombine those molecules to have the ideal polypharmacological, physical, chemical, and pharmacokinetic properties. For the ligand protein interaction, we have a technology called Matchmaker, which is an internally built deep learning engine that designs molecules to ensure the molecules are designed to interact with the right targets and avoid the wrong targets. And we have an internally built machine learning engine called POEM, acronym POEM, that ensures that the molecules are designed to have the right PK properties like blood-brain barrier penetration, hERG, oral bioavailability, etc. Next slide, please. The second platform that we have is Ligand Express. Unlike Ligand Design, Ligand Express is built to take a small molecule that exists and to uncover all the potential proteins that are predicted to interact with that small molecule. Next slide. It accomplishes this in a three-step process. It takes a small molecule, it evaluates all the proteins that are predicted to interact with that small molecule through Matchmaker in step 1. It then, for any given ligand-protein interaction, provides a prediction on modulatory effect, whether or not a molecule is predicted to have an antagonistic or agonistic interaction with a protein. And then it leverages a wealth of systems biology and genetic data to take a more downstream view on, which diseases is this relevant for? What about drug-drug interactions? And with the genetic information to provide insights into potential mutations at predicted binding sites in a way we call structural pharmacogenomics. So Ligand Design is for drug design, Ligand Express for off-target profiling. Next slide, please. And the application of Ligand Express is for target deconvolution. If you have a phenotypic screen where you have molecules that have lit up an assay or seen some biological readout, but the target is not known, the mechanism of action is unknown, Ligand Express is used to deconvolute the mechanism of action. It's also been used for drug repurposing, and we've applied Ligand Express in over 20 different partnerships with institutions across the world for COVID drug repurposing with some very exciting results, as well as in lead prioritization. Next slide, please. Now, when you think about Ligand Design and Ligand Express, we have one for drug design, we have one for off-target profiling. And when they're combined, we're able to design molecules as well as profile their off-target interactions, all in an integrated manner, all computationally and at the power of our computers. Next slide, please. And from a business model standpoint, we work with a vast majority of the big pharma companies globally, including a majority of the Japanese pharma companies with whom we have very deep relationships through collaborations, as well as we partner with early stage biotech companies, as well as academic institutions. And we've created over 12 companies to date and have ownership of over 65 different programs in preclinical across different therapeutic areas. And by partnering with early stage biotech companies and working with multinational pharma companies, our goal is to build the biotech pipeline of the future and to feed very solid science in preclinical into clinical through partnerships and to turn molecules into medicines. Next slide, please. And we are very active in this business model. We have relationships with big pharma companies all across the world. In the introduction, it was shown that we had done a deal with Yuhan Pharma in Korea. We recently announced a deal with Cerevel, one of the largest neurodegenerative companies, a spin out of Pfizer. We have relationships with Merck, and we just spun out a company with the University of Toronto that we'll be announcing before the end of the year. Next slide, please. And so with that, for anybody working on small molecules, for protein targets that are structurally represented, and you're looking for new chemistry, lead optimization, off-target profiling, please contact us. We'd be very happy to find a way to work with you to advance your research. Thank you very much.
Great, thank you, Nahid. Can I just quickly ask, would you be able to share these slides afterwards with the attendees? Or maybe a edited version, maybe?
No, absolutely. We'll get you these slides. And Tyson, I believe Arthur sent these slides. Feel free to send them over.
Okay, yeah, I have them. Perfect. Great, thank you very much. So thank you, and we'll come back for some Q&A later. And so next I'd like to introduce Stéphane Gagné, who's the president and CEO of Ovensa. Actually, we first met more than 10 or 15 years ago in life sciences events. So I'm really happy to sort of reconnect and showcase the company he's working with now. It's sort of interesting, more on the drug delivery side and slightly different approach. And I'm really interested to hear this, but I just want to, before I ask you to take the mic, Stefan, maybe the first thing you could do is just share if everybody, if you can share the slides with the attendees as well.
Definitely, yes, please.
Okay, great. Okay, thank you. So Stefan, I give you the mic.
Thank you very much, Tyson. So again, Ohayo gozaimasu, everyone. Hajimemashite. I'm Stéphane Gagné, President and CEO of Ovensa. I'd like to thank the organizers, Jethro and湘南Air Park, for giving us the opportunity to present today. Next slide, please. So Ovensa is a company that does drug delivery, as mentioned by Tyson. We collaborate with biotech and pharma companies to deliver their drugs more specifically across the various barriers. It can be the skin barrier, it can be the intestinal barrier, or the blood-brain barrier. I would say that we do more and more of the blood-brain barrier, the BBB, and we have our own program on immuno-oncology with a siRNA that targets galectin-1, which is highly expressed in a number of cancers, which is kind of a troublemaker, providing a lot of cancer resistance, and we're focusing on head and neck cancer. Next slide, please. So as you know, one of the main challenges with therapeutics is the difficulty to cross barriers. And if it does, again, there's a small percentage that once passed may affect the healthy cells, not only the targeted cells, but the healthy cells. And that's why, again, I think that there is some synergies with Cyclica using their ligands to be able to better target the targeted cells. And as well, some of the issues are that the therapeutics may be destroyed by natural defenses, by our immune system. Next slide. So what we have developed at Ovensa is a biopolymer that we call Triozan. Triozan is being used with high cationic charges for nano-encapsulating or micro-encapsulating one drug or multiple drug payloads. And we have the possibility to conjugate ligands on its surface that will make the drug more specific towards certain tissues or to certain cells. So as for example, in the case of where you take a drug orally, the Triozan will protect the payload from the gastrointestinal environment, have a strong adherence to the mucosa, open the tight junctions, getting into the bloodstream, and then bring the payload to the targeted cells. And via caveolae or receptor-mediated endocytosis, we can get the drug inside those targeted cells. Next slide. So we have the possibility, just like the key and lock analogy, to use a triazine to make it more specific towards certain tissues. So if you say, oh, we'd like to bring it more to the brain or to the liver, we have been working with pharmaceutical companies to do just that. So we can work with small molecules, but we can also work with biologics. Next slide, please. One thing also that we have done is to work on modulating the release of the payload. So it can be released within hours, or it can be released over 275 hours. That's the kind of range of that we have experience with the triazine releasing different types of payloads. Next slide. So as I said earlier, we are focusing more and more on crossing the BBB, because this is the holy grail in pharma. And we have used different routes of administration for doing this. So depending on the route of administration, whether it's intranasal, intravenous, intraperitoneal, intraarterial, the range or duplication of the penetration of the molecule in the brain varies. And we are working again with either small molecules. We've done things on autism models where we've been able to increase 33 times the ratio compared to in the brain and the hypothalamus compared to the plasma ratio. Next slide. So we have done some works with pharmaceutical companies that unfortunately I cannot present today because they are proprietary with intranasal delivery or IV. This one example here is something that is not specific with a partner that we have done, where we're getting a bispecific antibody in IV form. And the goal was to see how we could increase the penetration of this antibody into the brain. So if you look on the right side, you will see that with the triazine at the bottom image, we have been able to significantly increase the crossing of the BBB for delivery of this antibody and reducing the quantity that would be otherwise going to the organs. Next one. So you've heard there's a lot of research that's being done in immuno-oncology regarding checkpoint inhibitors still today. Unfortunately, still a small percentage of patients are responding to checkpoint inhibitors. This is now, thankfully to new research recently published, this is increasing, but there is a lot of course, mechanisms of action by which cancer cells defend or tumors defend themselves. Next slide. So what we are focusing at Ovensa is galectin-1. Gal-1 is really a troublemaker in cancer resistance. So it reduces the efficacy of radiotherapy, chemotherapy, induces T cell apoptosis, promotes angiogenesis. So the idea here is to reduce the quantity of Gal-1 by reducing expression with our siRNA, and that's what the efforts are today, is increasing the efficacy of checkpoint inhibitors and other anti-cancer treatments. Next slide. What we're doing is we're taking our triazine delivery system to deliver this siRNA, and we combine it with an anti-PD-1. And we have today, those are preliminary results, but very high transfection efficiency. Next slide. We have also been able to demonstrate safety profile of the siRNA. And what is important to see also is you look at on the right side at the bottom, you see that typically nanoparticles would accumulate in the liver or in the spleen, and we've been able to promote and target more specifically the delivery into the kidneys, the lungs, and into the tumor itself in the head and neck cancer model, and not into the liver or spleen. Next slide. And those are also very recent data where we show that when we combine our siRNA with an anti-PD-1, we're able to significantly reduce tumor growth by close to 50%. in 11 days. Next slide. What also we have done is work on recurrent glioblastoma. So that's a very tough cancer. As you know, prognostic are very, very bad with GBM. We've been able to get not only the sRNA through the nasal mucosa, but also into the olfactory bulbus and into the hindbrain. Next slide. But also we have demonstrated is to be able to bring not only into the tumor microenvironment, but we've taken this sRNA at the center of the tumor for better efficacy. Next slide. So that translates into significant results. So if you look at this graph here, the sRNA by itself is not as efficacious as temozolomide, which is TMZ. But when you give the sRNA prior to chemotherapy, you're able to reduce the quantity of GAL1 and then have a synergistic effect, translating into 40% median survival improvement and 40% long-term survival. Next slide. Also, when we combine with an anti-PD-1, then again, similar pattern, you see that the sRNA is not as good as the anti-PD-1, but when you combine the two together by giving the sRNA prior to the anti-PD-1, then we have another case of synergistic activity with a 42% median survival improvement and also 20% long-term survival. Next slide. So for the record, our delivery system Trialzan has been granted patents. So our patents have been granted in Canada, in US, in Europe, and also in Japan. We also have a proprietary targeting technology, which is a bradykinin-1 receptor agonist and antagonist, not only helping to better target cancer cells, but also we use copper-64 for brain imaging. Again, those patents have been issued and granted. And we also have our sRNA, which is our drug candidate, where again, the patents have been granted. Next slide. So just giving you a quick brief overview on our scientific advisory board. We work with top scientists. Quinn Shuley from Stanford University in the US is one of the top worldwide specialists in Galectin-1, so we work with her. She's a radio-oncologist. Dr. de Vluchauer, neurosurgeon, one of the top in Europe, who is behind this sRNA. And we work with other highly profile researchers, Dr. Gilles Bessin, who has worked at Moderna, Dr. Devan Bitzberger, who was the co-inventor of Baqsimi that was just acquired by Eli Lilly, and other scientists that are specialists either in cancer or in nanoformulations. Next slide. So what we're looking for in terms of collaborations, we are now raising $15 million US for our Series A that will close before the end of the year. So really active now into our Series A. And we're looking for additional collaborations. We had our first collaboration with Takeda Pharmaceutical that was signed last year to take an antibody to the brain, and that's public knowledge. That's why I'm mentioning. But more recently, we signed also R&D agreements with 3 more pharmaceutical companies in Japan, with which we will do active drug delivery. So if you're interested in a collaboration, our model is very easy. There's no fee to pay to access our technology for R&D, so we make it very easy for those types of collaborations. Next slide. And if you're interested, of course, please contact me. I'll be happy to, again, provide you complimentary information, published studies, and I already gave the authorization to share the slides.
Great, Stefan, thank you very much. Very good overview, and nice that you mentioned the partnerships you have already in Japan and looking to expand on that. That's great. So let me move on to the last speaker, Morris Smith. He's VP of Partnership and Product at Lumasonix. Lumasonix, she's a 2020 Bio Startup finalist. and they're focused on early stage cancer detection using photoacoustics and ultrasound imaging. And before we start, I just want to mention two things. One is, yes, Morris said that he's happy to share the slides after the attendees. And also, I'd like to just mention that Mr. Naveed Nazir, the CEO of Lumasonix, is also on the call, and he may join in the Q&A. Go ahead, Morris.
Okay. Next slide. Welcome to Lumasonix. We're in general cancer screening through blood imaging, and this would be fairly unique. There's been attempts using biochemistry to accomplish this, and we're purely based on physics-based imaging. Next slide. Thanks. Okay, so the problem is that 38% of people get cancer and around 19% die. And the period of development extends over a long period, could be well over a decade. And that gives potential for early detection, which is the single most important change to impact outcomes. There's been a lot of development on treatment, but that's fairly incremental. And it's thought that if we knew early that you have cancer, we could effectively treat it, even using fairly old therapies. So we're directing our efforts at early detection through general screening with a simple, reliable, low-cost, fast test that can be scaled for the whole country. And presently, there is no general screening test. Current efforts have used biochemical reagents for some specific cancers, but isn't practical for general screening. So there are efforts right now, but if you look carefully at them, couldn't apply them to the population of Japan, for example. And they also target a limited number of cancer types and are expensive, labor-intensive, slow, and damage the sample with the reagents. Next slide. Our solution is the Lumasonix cell analyzer, and we're using photoacoustics and ultrasound sensors, which will collect data on each cell in a blood sample. We have a manual process that we're converting through automation into automated screening, essentially an automated screening machine. An AI engine will classify healthy cells and determine the cancer type based on physical size and shape data. Circulating tumor cells are shed from growing tumors and cause cancer to metastasize, and they're well known as a characteristic of tumors. So we'll use photoacoustics and ultrasound imaging and our data analysis to count and classify the tumor cells and report them to the doctor. And that would be most valuable very early when you don't feel sick, and then they can effectively remove the tumor. And this would be the only system trained to recognize cancer from patient blood samples. So essentially, the way we know difference between regular cells and tumor cells is by actually looking at known tumor cells. And it can also be used for new drug trials or treatment monitoring and may improve data quality rather than using X-rays, for example, to monitor cancer and provide faster reporting, counting CTCs as opposed to the currently used alternatives. Next slide. We expect a 2 to 3 year development for the automation of the current manual process and generation of data. And the manual process is documented and published as university research. It's also patented. And we expect that by the 4th year after our development program that we should be self-sustaining and base our business on a fee split model with current blood testing labs, at least in North America. The technology development is right now on U.S. healthcare practices and going through FDA. And we have access to an accelerated regulatory process because of the nature of what we're doing. There's also government funds available in Canada where development takes place here. But we are open to developing in Japan if that's of interest. Next slide. Between normal cells and cancer cells, there's a variety of differences. The cancer cells can have variations in size and shape that are much different than normal cells, and the nucleus can be larger, darker, and various other types of physical characteristics. And using physical characteristics, we can separate the two types of cells. Next slide. This is a block diagram of our prototype manual cell analyzer, and it uses the two types of imaging, photoacoustics, which we would introduce to medicine, and ultrasound to detect the size and shape data of the cell. So we would go through the cells one by one and classify them as a red blood cell, for example, or a tumor cell, and then identify the tumor cell type and count them up and tell the doctor. So essentially flagging CTCs in the sample for the doctor to do follow-on ordinary testing. Next slide. This is a picture of the manual prototype, and you can see the flow device for cells and the laser and CCD camera we use, and also the photo acoustics on the left, and then there's a blow up on the right of the flow channel. Next slide. This is an example of the type of data that we can, and the photoacoustic is from the nucleus, and ultrasound imaging from the cytoplasm. And then there's two types of cancer cells shown, breast cancer and colorectal cells. And you're looking at, on the right-hand side, the clustering of the different types of cells, which is quite different from normal cells. So the red blood cells and these two types of cells would be quite different. Next slide, please. This shows the two types of imaging again, and we have red blood cells, white cells, and melanoma cells. And by using the two imaging types together, we can separate the cell types. Next slide. This is scatter plots for cancer and normal cells, showing inner diameter and circularity by volume. And this is for a wide variety of cells. It wasn't done by this present technique, but it shows how the physical characteristics vary. And you'll note that the normal cells are with diameters below 10 micron, and then the tumor cells are on the right-hand side, which corresponds with diameters bigger than 10 micron. And there's particular characteristics like fingerprints for each type of tumor cell. And so we'll read those in from the patient blood samples where we know the type of cancer, and then match an unknown sample to the pattern of the tumor cells. Next slide, please. The competitive advantages are we work without the fluorescent reagents, and that's common to any biochemical process, whereas because we're purely physics, we're simply looking at the actual cells physically. And so we don't need the fluorescent reagents. That also makes it something we can automate, and we don't damage. And so we're looking at each cell individually, delivering the high sensitivity and specificity. The cancer biomarkers are not needed, or we don't rely on the presence of specific cancer biomarkers. And we can look at the full distribution of cell mutations. and essentially increase sensitivity by looking at more blood. So the choice of sensitivity depends on how many cells that you look at and can be increased to the point desired. We've referenced 10 milliliter sample, so that's the standard sample size, and that should have sufficient sensitivity based on, there's been studies of the amount of cancer tumor cells in a standard blood sample. So effectively, we can look at the early stage that way. And we're not destructive, so you won't even need to have extra blood drawn and can use the same blood if needed for other test processes. Next slide. So we're intending to have a fully automated classification count of the tumor cells and then fee split in the US with major lab providers. And we've modeled it at a $100 patient fee and split with the test provider. And normally people have annual checkups, and this would just fit in with their annual checkup. And the labs do it because it increases their net margin and provides them a lot more revenue. And we don't need insurance payment, but they would want to shift to early stage care. So everybody, every stakeholder is aligned with this, and we would simply place and maintain machines at the labs. Next slide. And we're looking for between $2 and $15 million to get through this process, and $2 million, that was the next stage prototypes, and then 15 million would go right through FDA approval. So essentially to the point where we're in production. And there's a variety of global patient screening opportunities. We're not going to be involved in the actual screening, and we'll be placing machines at current blood testing labs, or someone could start that in the country. And there's manufacturing opportunities, and this could be expanded to test cats and dogs, which have a different viscosity, but it's a similar pattern of detection. And there's also general lab applications, because we can detect other types of cells that are differentiable based on size and shape data. And the cost of the business beyond development would be generally by bank financing. Next slide.
Morris, we're going to need to get you to wrap up pretty quickly.
Okay.
Thank you.
Okay, so I think that's the last slide, right? I think we're done.
Perfect.
Okay.